Well, good morning, everyone. I'm really excited to be here today to share with you how we've used Google Cloud Platform to analyze genomic data. So a little bit about myself. I'm Christine, and I lead a team of software engineers and bioinformaticians at Myriad. As a hybrid team, we're not just interested in analyzing genomic data, but we're also interested in choosing the right tools to do so, especially in a clinical lab setting of our size. So first of all, who are we? We are a genetic testing lab with a mission to deliver genomic information to patients at each stage of their life. So this requires a deep understanding of technology across many different domains. So for example, we need to have a deep understanding of genomic data to understand how to choose the right tools to gather and structure genomic data, interpret it, and then ultimately deliver the right information to patients. So that's kind of from a mission standpoint. From a product standpoint, we have 3 different products for delivering genomic information to patients at different stages of life. So our first test is our carrier screening test. This is for couples who are planning on getting pregnant and want to know if they are both carriers for a recessive disease. So we can screen for over 200 inherited recessive conditions, including cystic fibrosis, spinal muscular atrophy, and Fragile X. Our prenatal test is for women who are pregnant, and this test detects fetal chromosomal abnormalities as early as 10 weeks into the pregnancy. This replaces sensitive ultrasounds and also invasive amniocentesis. And finally, our inherited cancer screening test is for men and women, and it tests for BRCA1, BRCA2 related breast and ovarian cancers, as well as several other types of inherited cancers. So, to get a sense of scale, this is a broad market with growing demand. There are currently 4.2 million pregnant women in the United States annually, and we've really only reached the tip of the iceberg in terms of delivering genetic information to them, with 1/5 of them receiving carrier screening tests and 1/3 of them receiving non-invasive prenatal testing. The market for inherited cancer screening is even larger, with more opportunities for growth, with 19 million women who meet criteria for inherited cancer screening and only 1.5 of them actually receiving preventative testing. So how do we do this? We're a clinical lab, and how do we actually test these patients for these different diseases? So it turns out like the typical flow of a sample is actually quite similar across all of our products. We essentially receive a tube of blood or saliva from the patient, and then we sequence the sample in our lab. Depending on this product, we sequence, we will do either high coverage targeted sequencing or low coverage whole genome sequencing. After sequencing is complete, we align the reads to the reference genome and look for variants in that sample's genome. After going through QC, it then moves into the variant interpretation phase. Variant interpretation is the step that is to determine which variants are disease-causing or not. Not all variants are actually disease-causing, and so we need to find the ones that are and actually calculate risk as well in this step. So finally, once we have all of the interpreted information, we are ready to send the report to the patient. So in this talk, I'm going to focus on how we use GCP for the problem of variant interpretation. So first let's talk about some of the challenges of variant interpretation. The challenges of variant interpretation come in 2 different flavors. One is the scalability flavor, and then the other is the diverse set of information that we need to interpret variants. So to really understand the challenge of variant interpretation, we first need to understand the scale of the problem. So how much data are we dealing with? So as Myriad, we are the largest molecular diagnostics company, and we have tested over 2 million patients to date. All of our tests are focused, which means that we test specific genes and diseases that we have predetermined are actionable. Each product, we test a different set of genes and diseases, but in total, we test for over 200 genes and diseases. So what does this mean in terms of base pair sequenced? Again, this depends on our product, but we can think of, on average, sequencing around 1 million base pairs per person. Some of our tests do low coverage whole genome sequencing, and some of our other tests have a smaller region of interest, but on average, we will sequence around 1 million base pairs per person. So on the database level, what does this mean? We need to keep track of all of the regions sequenced in a person. So to kind of like blow that into perspective, we want to keep track of the 1 million times 2 million base pairs that we've ever sequenced in the lab. We also want to keep track of the variants that we see per person. So most of the time, the genomic sequence of a person is the same as the average person. Variability in humans are estimated to be 1 in 1,000 base pairs. So a naive database representation of the number of variants that we see per person would be 1 million divided by 1,000, which is 1,000 variants per person, multiplied by the 2 million patients that we have tested, or 2 billion rows. So that's that gives you an idea of the amount of sequencing data that we need to keep track of. For variant interpretation, we want to know what the number of novel variants are, because we want to determine for each variant whether or not it's disease-causing or not. So this graph just shows that even though you've been testing for a while, there is a long tail of rare variants that exist. A vast majority of these variants are benign, but a small percentage actually break the function that they are in, and the problem of variant interpretation is to determine which one of these are disease-causing or not. So this kind of gives you an idea of the scale of the problem, but we need to understand the other challenge, which is the many different factors that are used to determine the classification of a variant. So for each novel variant, we need to determine if it's disease-causing or not by gathering and weighing different types of evidence. So you don't actually have to read this, but these are guidelines that are produced by the American Society of Medical Genetics that just show the multiple types of evidence that one needs to consider when determining the classification of a variant. I'm going to just highlight one of these types of evidence, which is allele frequency. Knowing the allele frequency can really help determine the pathogenicity of the variant, because if we see this allele occur more often than the disease itself occurs, then it is less likely to be a pathogenic variant. And so you can classify this allele as benign if it has a high allele frequency. So what is allele frequency? It's pretty straightforward. It's calculated as a fraction, the number of times that we see an allele in a population over the number of times that this particular locus of the allele was sequenced in the population. This is fairly straightforward, but just want to highlight that we need to parse a lot of genomic data to get this calculation. So we need, for AC, which is allele count, we need to go through all of the calls of every sample to calculate this. And for AN, which is the number of times that we've sequenced a particular locus, we need to go through all of the regions sequenced in every single sample. So that's a lot of genomic data. Another type of evidence is zygosity count. So zygosity count is similar to allele frequency in that it's counting the number of times a certain genotype is seen in a population. So since humans are diploids, we have 2 copies of each of our chromosome. One is from our mom and one is from our father. So most people are the same as the reference genome, and so that makes them homozygous for the reference sequence. Some people have a mutation on one copy of their chromosome, making them heterozygous. And sometimes a person has a mutation on both copies of their chromosome, making them homozygous for the alternate sequence. And in some rare cases, a person actually only has one copy of the chromosome at a particular locus, making them what we call a hemizygote. So for variant interpretation, we're really interested in knowing the zygosity count because if several people are homozygous for the alternate sequence, then that makes this allele less likely to be disease-causing. All of these questions really fall under a class of analyses called tertiary analysis, where we try to make sense of sequencing data. So just to contrast between primary analysis, which is really trying to interpret the data that's coming off of the sequencing machines and output sequencing reads, and contrast with secondary analysis, which aims to assemble and align the reads to a genome and call variants. Tertiary analysis tries to make sense of this variation in a biological way. So other types of questions that we may want to ask when we're doing tertiary analysis would be, how often do we see a combination of alleles together in one patient? We also might want to calculate these metrics on different segments of the population. So we may want to segment by ethnicity, for example. Sometimes an allele can have a high frequency in one ethnicity, but not others, and that would give it enough evidence to classify it as benign. We may also want to segment by gender, because there are some diseases like cystic fibrosis that have different outcomes per gender, and so you might want to look only at the female population if you want to assume a healthy population. Again, we always, when calculating these metrics, we do want to assume that we have a healthy population, so we potentially want to sequence out any patients with any sort of testing indication that's not routine carrier screening. That means that they might be getting tested for a particular reason, and they are probably actually not considered to be part of the healthy population. Furthermore, for research purposes, we really want to make sure that we are only including samples that have consented to research and are in research-allowed jurisdictions. So this is another type of segmentation of population that we are interested in the research realm. Finally, tertiary analysis does not need to be limited to variant interpretation. We may, for example, want to infer ethnicity of patients based on their genetic information, and this falls under the realm of tertiary analysis. So for a lot of these questions, we can utilize public databases. We can see if the variant has been observed in that particular population and look at what the allele frequency and the zygosity are in that population. But some caveats are that these databases are often limited in size, which means that rare variants are less likely to be observed, and if they are observed, they may have a good chance of being overrepresented. Furthermore, we aren't actually able to answer some questions like co-occurrence without knowing the genotype at an individual level. So we do use these public data sets, but these are just like one type of evidence that we consider. Really, in order to provide the best information to our patients, we have to leverage our own internal data set of over 2 million patients. With this larger population, we have much more statistical power for variant interpretation, and we can also go beyond variant interpretation and contribute back to science and do our own retrospective analyses. So again, when we're doing research, we have to be very careful in making sure that our samples have been consented for research and are in research-allowed jurisdictions. But once we have that population, then we can, for example, ask questions like, what is our carrier rate, to understand more of the efficacy of expanded carrier screening, especially in a diverse ethnic population. So what is the carrier rate? What is the at-risk couple rate? Those are all very interesting questions to basically prove efficacy of our tests. With this platform, we can also observe other patterns, such as coupling patterns across ethnicities, and we can observe many other types of these analyses with the data that we have. So hopefully this motivates building a system for tertiary analysis that requires tackling two major challenges. The first one is scalability, and the second is flexibility. We need a flexible system because, unlike clinical trials or other hypothesis-driven projects, the set of metadata that might be relevant for analysis is diverse and potentially unknown upfront. So for example, I could ask to see all of the homozygous individuals that have a co-occurring pathogenic mutation. One group may want to see a certain set of metadata for variant interpretation, and another group may want to see another set of metadata for retrospective analysis. Furthermore, different groups may have different restrictions and access to data. So we really need a solution that allows for flexible ad hoc aggregation and filtering of variant information along with sample metadata. We also need a scalable system. We have a very large population data set, and this population data set is growing every single day. So unlike some of the public databases or academic cohorts, this is a patient population that is growing every single day, and we need to calculate these metrics on the latest information. So I just wanted to talk about one implementation that we had before using BigQuery. So we essentially hand-built a batch compute with Luigi to calculate allele frequencies. This pipeline was very optimized for the one metric that it needed to calculate. We basically took advantage of the fact that this was a recursive procedure and stored the previous day's AC, AN, AF, and would add to that as needed as we got new samples every single day. But some of the problems that we ran into was that it was not flexible. So if we wanted to add another metric, we had to build a completely different pipeline. And these would often take weeks to run on all of our historical data. We could obviously increase the instance size and the compute power, but it would still be very compute intensive. The other thing is that it's really not able to do ad hoc aggregation and filtering. You really have to be thinking about what is the information that you want up front, and then building the pipeline to calculate it. So it's not a very flexible system. It's not scalable in the sense that we aren't able to really generate these metrics in an ad hoc way with the amount of data that we have in the time that scientists need to see them in real time. So that kind of motivates why we decided to look into using BigQuery. So this is just a very high-level architectural overview of our system. We have genomic data and sample data that is stored on Google Cloud Storage. This is ingested into BigQuery using Dataflow, and then right now we have just a very simple system that persists the results back into cloud storage. So as new data comes in, we add new rows to BigQuery. So let me just go into each of the individual steps. So this is the input data that we're dealing with. We store genomic data and sample data. I will go into a little bit more detail about what the genomic data looks like. The sample data is kind of anything that you might want to know about the sample. So that would be gender, ethnicity, the testing indications, family history, et cetera. And for the genomic information, we store this in a format called GVCF. So if you work with genomic data, you may be familiar with the concept of VCF, which is the variant call format. This is a file that basically contains all of the call information for a particular sample. A genomic VCF is very similar to the VCF, but it also includes the homozygous reference regions. So essentially, this allows us to distinguish between regions that were sequenced and regions that were not. And so you can see that there are large blocks of homozygous reference regions in this particular sample that indicates that this person was sequenced here, but no variants were called here. So then, so we have all of this information, genomic information and sample information in our Google Cloud Storage. The next thing we want to do is transform and load this into BigQuery. So the way that we chose to do this was we chose to parse the GVCFs and transform them into a Parquet format. With this, we're able to really be flexible about the schema that we ultimately want in BigQuery, and we're able to. And then at the end of the day, we have these Parquet files that we could potentially use in other areas, in other cloud systems as well. So we parse our GVCFs, every single sample, transform them into Parquet, and then append the new samples to our BigQuery table. And similarly, we do this with the sample metadata. So this is an example of what the BigQuery table ends up looking like after we load it in with Dataflow. As you can see, it's very similar to the GVCF that I showed earlier. It has your chromosome position, ref alt, and the genotype information available. So now I'll just kind of go, this is kind of a high-level overview of a sample query. So to calculate allele count, we split this up into two steps, really. The first is to parse the genotype field so that every single row represents a single allele for a sample. And then after we do this, after we flatten the table, we are able to aggregate all the alleles together to generate the allele count. And so this is kind of so this is like a simple group by, and then a count, an aggregate. So I wanted to talk a little bit about some of the tricky cases that we have dealt with, because on the surface level, this may seem like very straightforward to do. But there are some tricky edge cases that we have to deal with, and one of them is the deduplication of samples. So there's a possibility that patients can get tested multiple times in our lab. And so as a result, there can be multiple samples that are associated with a particular patient. And we want to use the most recent sample because those are assumed to be the most accurate. In our previous pipeline implementation, we had to do this by calculating since we were calculating the metrics incrementally, we had to basically subtract out the counts of the samples that were duplicate, and append that subtraction to our final allele count. But with BigQuery, you can kind of, it's like much easier to deduplicate the samples up front and then kind of recompute your metrics. So the other tricky case that I wanted to talk about is the deduplication of alleles in BigQuery. We often define, there can be multiple representations of the same allele. So when working with genomic data, we often define alleles in, like according to the reference sequence. But as you guys may know, there are many versions of the reference genome. And even with the same reference genome, there are potentially multiple representations in very repetitive regions. So this particular example is an example of an insertion deletion, an indel, in a repetitive region of the genome. And you can see that there are potentially 4 different representations of this allele when compared to the reference genome. So this is a known problem in the field of genomics. There are many working groups out there, one and the Global Alliance for Genomic Health, that are dedicated to solving this problem. So how we've decided to address this problem is very similar to the way that Global Alliance for Genomic Health has also addressed it, by using a content addressable hash. So this is an internally built library that we built that is used to tag alleles up front. So then we'll tag them with the hash, and then it makes it much easier to combine alleles later in the BigQuery pipeline. And then so finally, the real advantage of BigQuery is this continuous ingestion of data. We're able to essentially just add rows to the BigQuery table as we see more samples coming through our lab. And because it's so easy to query on this, we can just recompute our metrics on the entire table. So how do we actually add more genomic calculations in BigQuery? It's as simple as writing another SQL query. So we can do this and done this for co-occurrence, zygosity, and dependent samples. Furthermore, putting it in BigQuery allows us to do a lot of ad hoc filtering and segmentation. We can filter and segment on ethnicity and gender, testing indication, et cetera. And so this really allows us the scalability that we were looking for and the flexibility that we were looking for in terms of ad hoc filtering. So to kind of wrap up, I wanted to talk about application in cystic fibrosis. Cystic fibrosis is one of the most common autosomal recessive conditions according to ACOG. It's caused by a mutation in the CFTR gene, which codes for the CFTR protein. CFTR protein acts as a channel on the membranes of the cells to control for the flow of water and ions in and out of the cell, especially in the lung. So if there's a pathogenic mutation on both chromosomes, this will cause cystic fibrosis. It does this by basically reducing the flow of these ions, and as a result, there's a thick mucus that tends to form on the surface of the cells. So this causes lung infections and limits the ability to breathe over time. So as a carrier screening lab, we have the advantage of having a very large healthy population. So with this very large healthy population, we are actually able to use our internal data to reclassify some variants that we previously did not have enough evidence to classify. And as a result, they were considered variants of unknown significance. Variants of unknown significance are just variants that we've exhausted all the information we know about this, but we still don't know whether or not it's disease-causing or not. These types of variants makes it really difficult to counsel patients because it's obviously better to know definitively whether it's benign or pathogenic. So this particular variant, we wanted to know how many patients had this allele along with another pathogenic variant. If this particular allele occurs very often with another pathogenic variant, it's much less likely that this is actually a pathogenic variant because the fact that we have a healthy population means that these people are alive and well and they don't have cystic fibrosis. So we asked that question, and it turns out that there were quite a few people that had a co-occurring mutation with a pathogenic mutation. But then we wanted to go a little bit further. So cystic fibrosis is an interesting disease in that there is a variable phenotype for males. And so it's very likely that there may be males with cystic fibrosis that don't even know it. And so to filter out the males and just look at the number of co-occurring pathogenic mutations in a female population, that gives us a much stronger statistical power to classify this variant as benign. And ultimately, we were able to do that. We were able to downgrade this variant to benign. This is another example where we actually did not have enough information to downgrade, so we asked the question, how many patients has this co-occurring mutation, has this mutation co-occurring with a pathogenic mutation? And the conclusion was that there was not enough to downgrade. And then finally, for this particular variant, we also were able to reclassify it, but we actually reclassified it from pathogenic to VUS. Given the answers to some of these questions, we actually think it's probably benign, but the fact that we're not giving a false positive to a patient is actually very, is better than that. So to downgrade this variant, we actually used both allele frequency and co-occurring statistics to downgrade. So it turns out that this allele actually occurs quite a lot in a healthy patient population, so it's considered to be very strong candidate for actually being benign.